Do you know what is the structural and functional unit of life? Yes, it is a living cell. And why it is considered the structural and functional unit? All the metabolic and regulatory function of an organism are carried out by the cell. That's why the cell is known as the structural and functional unit of life. Have you ever wondered how it is possible to carry out all functions in this tiny structure? Yes, it is through cell organelles. They are specialized structures present inside the cell having specific functions. Let us discuss these structures one by one. All organisms need energy to survive. A cell also requires energy for their proper functioning and maintenance. So, how do they get energy? The cell gets its energy from a specific cell organelle named mitochondria. It is commonly known as the powerhouse of the cell and it is the site for aerobic respiration. Mitochondria is a sausage-shaped membrane-bound organelle having an outer membrane and inner membrane. These membranes divides its lumen into two aqueous compartments, the outer compartment and inner compartment. To increase the surface area, the inner membrane contains a number of infoldings called cristae and it is filled with a homogeneous substance called matrix. For protein synthesis, the matrix contain single circular DNA, ribosomes and RNA molecule. So we can say that mitochondria are a semi-autonomous organelle in the cell, which means mitochondria possess their own DNA and ribosome unlike other cells. The number of mitochondria in a cell depends on physiological activities of that cell and the number of mitochondria increases by fission. As we discussed earlier, for protein synthesis, ribosomes are required, right? The ribosomes the only organelle present in both prokaryotes and eukaryotes. So, do you think prokaryotic and eukaryotic ribosomes are the same? The non-membranous, granular structured ribosomes were discovered by George Plastids in 1953. It is composed of ribonucleic acid and protein. Structurally, ribosomes consist of two subunits, a larger and smaller subunit. So, the prokaryotic ribosomes are 70S ribosomes consisting of 50S larger subunit and a 30S smaller subunit. Here, the S indicates Swedberg's unit. It is an indirect measure of density and size. The only difference between prokaryotic and eukaryotic ribosome is the difference in the Swedberg unit. That is, the eukaryotic ribosomes are 80S ribosome, which is a combination of 60S larger subunit and 40S smaller subunit. Now let's focus on another organelle, the plastid, which are large pigmented organelle found only in plant cells. As mentioned before, plastids bear some specific pigments. Based on the type of pigments present in it, they are classified into three. Let's discuss the types of plastids and their functions in detail. We are all familiar with the pigment chlorophyll and carotenoid. They are responsible for photosynthesis by trapping sunlight. Both these pigments are present in chloroplast. The majority of chloroplast is present in the mesophyll cells of plant leaves in variable shapes. Do you know, in Chlamydomonas, only one chloroplast is present in a cell. But generally, in the case of green algae, 20 to 40 chloroplasts are present per cell. So we can conclude that the number of chloroplasts varies according to the plant species. From the image, you can understand that the chloroplast is a double membrane bound structure like mitochondria. Inside the inner membrane, a colorless fluid is present and it is called the stroma. Do you know one thing about the stroma? It contains specific enzymes for the synthesis of carbohydrates and proteins. Inside the stroma, you can see several membranous flattened coin-like structures called thylakoids. They are responsible for the occurrence of light reactions during the process of photosynthesis due to the presence of chlorophyll pigment. Here, you can see that thylakoids are arranged in a stack-like manner called grana. About 10 to 100 grana are present in a chloroplast. When you observe the image, flat membranous tubules connect each thylakoid by stroma lamellae. In addition to these structures, double-stranded DNA and ribosomes are present in the stroma of the chloroplast. Now let's move on to the another type of plastid, namely chromoplast. As the name suggests, it contains colored pigments like carotene, xanthophylls, and other red pigments. These pigments are responsible for the orange, yellow, and red coloration of plant parts. Here is a question for you. Why the carrot and beetroot are in orange and red color respectively? Yes, due to the presence of fat-soluble carotene and xanthophyll. So, now we are moving on to an exceptional type of plastid, that is the colorless plastid, leucoplast. What is the function of leucoplast? We can assume that the function of the leucoplast is entirely different from the function of chloroplast and chromoplast because it is a colorless plastid. Do you have any guesses about its function? Yes, the leucoplast is involved in the storage of carbohydrate by amyloplast, fats and oils by eleoplast, and proteins by aleuroplast or proteinoplast. So, where the leucoplast is present? As you know, they are colorless plastid. So, they are present in non-photosynthetic parts of plant like roots, endosperm and tubers. Is there any machinery or any mechanism to control and regulate all these processes inside the cell? Let us find out in the next video. Now let's summarize what we have learned.
Manorama Horizon. Learning made simple.